Thank you, everyone. Thank you for the wonderful invitation to speak in this fantastic series. And thank you for all of you here in attendance and those online. As you'll notice, I have a little bit of an accent, so I'll try and speak slowly and clearly. But you're welcome to get clarification afterwards, lots and lots of questions. I would also like to start my presentation with my acknowledgement slide to recognise all the wonderful people who've contributed over the years significantly to the work that I'm going to be talking about. And so this includes all my fantastic clinical colleagues, which through some of this work's not going to be possible. Obviously all the patients for participating in research, which is really, really essential. But also members of my team that have contributed in various ways, and particularly for the work I'm going to be focusing on, particularly at the last half of the presentation, is work primarily done by Ryan Anderson with the significant contributions also from Makiko and Lisa. And also acknowledge some of the funding that supported the work that I'm going to be talking about as well. So I think it's fair to say that, regardless of your educational backgrounds, whether you have a science training, a medical training, or none of that, that we all recognise that both genes and diet, certainly play a role in adult chronic diseases. So we talk about whether we have a family history of particular conditions, whether it's hypertension, heart disease, or diabetes. But we also know when we have those conditions that dietary modifications can particularly improve the outcome of those conditions or stave off that disease as well. And so with that, when we turn to other types of disease, in this case here, structural birth defects, and so structural birth defects are talking about these are physical abnormalities that often arise in the first trimester of gestation, so embryonic development. Here, we're sort of less sure about the relative contribution of some of these dietary factors. Just to give you an idea of the significance of structural birth defects, or overall birth defects, about 31 million kids born per year with birth defects. There's about 1 in every 33 in the US. Now, many of those aren't severe major birth defects, but there are a significant proportion of them that are. And of those, genetic contribution is well recognised. But also from epidemiological studies, we do know that things like nutrition and lots of environmental exposures also contribute to those risks, although it's far less well defined than it is for adult chronic diseases. And most of these are really based on statistical associations, looking at large human populations where there's a huge amount of variability. The one exception I think that everyone's probably heard about is that of folate or folic acid, which is vitamin B9. And so it's widely recommended, and probably most of you, particularly the women in the audience, but hopefully most of the men also realise that this is recommended in preparation for becoming pregnant and also during the early stages of pregnancy as a way to help prevent or reduce the chances of being born with a neural tube defect such as spina bifida. And so spina bifida is this failure of a complete enclosure of the neural tube during development. This occurs about in the 4th week of human development, so well before many women know they're pregnant. And this was really based on the seminal findings from clinicians in the 70s and 80s, where they identified low serum folate in about 70% of pregnant women who had children born with spina bifida. But it really wasn't until animal studies could actually go on to support that there was a functional correlation, so you could actually get supplementation with folate in the diet, and that would actually help mitigate or prevent some of the severity of these defects. And here, just showing some examples, and these are actually images that we've taken in collaboration with a couple of other teams that are working on this particular birth defect, looking at even more effective ways to treat some of these conditions. And so these are mouse embryos with spina bifida on there, and you can see it has a significant effect, not just onto the outside of the embryo. So folate actually has multiple roles within the cell. It contributes to the synthesis of the building blocks of DNA and RNA. It also regulates the metabolism of a natural amino acid called homocysteine, which then in turn feeds in to its contribution of methyl groups to regulate gene expression across the genome. So that's where most of the effort is focused on the contribution of folate in terms of regulating gene expression. But despite the success story of folate substitution has really been shown to, it helps prevent the condition, and certainly from decades of now studies in humans where we have fortification of foods, we still have a significant decrease in the incidence of spina bifida, but it doesn't actually eliminate it completely. And animal studies suggest that about a quarter of the genetic causes of spina bifida do not respond to folate. So a lot more work is still needed on successful examples like this. So I'm going to switch now to really what my lab does as a primary area of research, and that's looking at craniofacial development, and particularly common craniofacial malformations or diseases. And so we tackle it from a variety of different approaches: human genetic studies, we look at molecular and cellular biology studies, we do a lot of animal modelling and quantitative imaging, and really to address what I'm going to be talking about today, some of the specific gene-diet interactions that can help potentially these sort of common conditions. So the main common facial birth defect that we focus on is cleft lip and cleft palate. Most of you are going to be familiar with this condition. I'd be surprised if many of you don't know someone or know of someone who was born with a cleft. It is the most common facial birth defect and one of the most common human birth defects in general, with about an average of about 1 in 700 live births, although it does vary quite significantly from different ethnic populations, suggesting that there's very distinct genetic contributions in these populations. Totally, that works out to be about 250,000 kids born every year with clefting. And just to put that in context too, that's about 3 times the number of children born with any form and diagnosed with any form of childhood cancer. So it's widely recognised, and those people who don't know someone personally will also might recognise many of these faces who are also born with cleft lip and cleft palate. So it's widely recognised, but what they actually go through in terms of the clinical treatment is perhaps not so well appreciated. And so we've got a number of questions we're really interested in addressing. What determines an individual's risk? What does drive some of the disease presentation? And there's a lot of variability that I'll talk about. And can we exploit this to understand potential interventional treatment options? So I want to break down to you some of the genetic contributions to facial development. I like to think about it in 3 different ways, and I want you to think about this as I go forward too. There's 3 different genetic contributions to facial variation, which also contributes to susceptibility to these disorders. So the first is the population level genetic variation. And so we can appreciate this when we look at sort of like average faces from different ethnicities. And those of you who travel a lot internationally will also probably pick this up too, that certain racial groups or certain countries have very particular craniofacial features. And so that's the population level genetic variation. So normally often benign things that just lead to differences in craniofacial shape. And then there's the variation that we all appreciate. So when a child is born, the first thing to say after they're cute is that they look like mum or dad. And that's your family-specific genetic variation that's contributing there. So that contributes to facial shape differences as well and features. And then you have these larger genetic variation, larger biological impact genetic variation. So mutations in single genes that give rise to changes in craniofacial morphology that superimpose on family-specific variation, but also population-level variation as well. And so these are these syndromes because they have this characteristic facial appearance that sort of overrides all that other variation. And so when we're thinking about genes and how the face is formed, there's a number of key cell types that drive this and a number of key events that occur that I want you also to keep in the back of your mind as we talk about clefting. So first up, the process of facial development really occurs primarily over this 4-week period. You see this massive growth of tissues at the anterior end of the embryo. And this is undergoing lots and lots of morphological change as well. And so this growth is actually driven by cells that are called mesenchymal cells. They're loosely connected cells. They're highly proliferative, and they're just growing and proliferating the whole time and growing out that face. You can see the face is actually doing a lot of other things there in terms of changing its shape and morphology. If you look from front on, these embryos here, you can start to see invaginations on either side, and then you'll start to see tissues start to join back together. And so this here ends up being the upper lip. You require fusion of these tissues to form the full upper lip. And these invaginations end up becoming the nostrils and give the full patency of the nasal airways. And so this is what goes wrong in patients with cleft lip and cleft palate. Just to show you this whole process, regardless of vertebrate species, is highly conserved. And so this is a mouse. This is humans at about 42 days of gestation. Mouse at about 11 days, the same thing occurs. The same invaginations, the same fusions. Chicken embryos do the same. They do it in about 5 days. The differences there are just the timing and the extent of growth of each of those facial processes. And they fused exactly the same way, and most of the genes are exactly the same. In terms of the roof of the mouth, the other part actually occurs about 3 or 4 weeks later, but it's derived from much the same sort of tissues as well and involves a fusion event as well. So I talked about mesenchyme and growth, but you saw lots and lots of morphological change too, or shape changes. And so this is actually driven largely by a very small population of cells, of epithelial cells that actually cover the entire embryo. But particularly in this facial area here, the epithelial cells at the tip of these processes actually provide the growth factors that drive the outgrowth of those mesenchymal cells and the proliferation of them, but also mediate the fusion of those processes. And then they disperse to allow the mesenchyme to come across so that bone can form fully across the lip and hold your teeth in place. And so this whole event here is dictated by massive changes in cell morphology, which is mediated by proteins that stick these cells together and how they're controlled. And so you've got this whole process of rapid growth, rapid morphological change, and this is a reason why I think that cleft lip and palate is really so common, because there's lots of little genetic insults and also environmental stresses that can actually disrupt that process even subtly. So one of the things I really want to stress about clefting though is this huge variability in clinical presentation. We always think about faces as being perfectly symmetric, but we're not. And so although you get lots of cases whereby you'll have a bilateral cleft lip and palate, often they'll have some asymmetry to them, but also frequently you might just get a failed fusion on one side of the face and not the other. And in fact, left-sided unilateral clefts are twice as common as right-sided clefts. And we'll come back to that. You can get, in severe cases, as I said, because the tissues are derived from the same embryonic tissue masses, that in severe cases you'll also get the roof of the mouth failing to close and fuse as well. And you can also get independently failed closure of the roof of the mouth, and then a whole lot of subclinical phenotypes. So this is a huge spectrum to deal with. And this has real impact for the patients themselves. So from a surgical perspective, over the decades, they've tried to standardise the treatment of care for a lot of these patients, and this is sort of like a typical plan of treatment where you'll see an early surgery, but not till 3 or 5 months of age, because you have to accommodate this massive growth that keeps going on in the neonatal period of patients and throughout childhood. And so you've got staged surgical interventions to deal with different things. And then you've got the orthodontic treatment and speech therapy. And so on average, the more severe the presentation, the more surgeries you'll need. In fact, I know a patient who had bilateral cleft lip and palate walked into my room. She'd had 24 surgeries, and I could still tell when she walked in the room that she was born with a bilateral cleft lip and palate. So it does depend on a lot of other factors too, and experience of the surgeon. So coming to somewhere like Children's is great because you've got fantastic surgeons here. In terms of how they present in the population, I said a lot of these cases, or 70% of these cleft cases, are isolated or called non-syndromic cases. So it's the only presentation in these kids is the cleft lip and cleft palate. And so often you will find them, there could just be the first appearance of that. So this is these are pedigree diagrams for those who aren't familiar with them, or family trees. Squares are males, circles are females, black labels are affected individuals. And so here you see a single affected individual with no family history. Other cases you'll see what's quite common is you'll see a distant relative who's affected, but many in between who aren't. And then in about 10% of cases you'll see families like this, where you'll see a much higher incidence of clefting in amongst the families. And so we think these are more likely to be significant single gene contributions than others. But even so, these still show incredible variability in presentation, that every individual will have a slightly different presentation. And obviously many people hear that it skips generations as well. And this sort of more closely resembles what we call the syndromic forms of clefting, and these are forms of cleft lip and palate that are associated with other recognisable features, and that collectively they present together. And so an example here, Van der Voort syndrome, which is caused by mutations in RF6, will have clefting, but will have lip pits in the lower lip, or pits in the lower lip, which are diagnostic of that syndrome. This one here, again, you'll have ectrodactyly or limb defects, eye defects, scalp defects. So they define particular syndromes. And so all these are caused by single gene mutations, or genes in a single, mutations in a single gene. They show these very classical dominant inheritance patterns where every individual might be affected who carries a mutation. But again, there is significant variability in amongst these cases. So if we come back to the non-syndromic ones, which are the more common ones, what goes on with the genetics there? So over the years, many decades of genome-wide association studies have identified about 45 common population-level genetic loci, or that are pinpointed genes in those regions that contribute to population-level risk. But collectively, even if you look at all 45 of these, collectively it's only contributing about 30% of your risk of being born with clefting. So there's a lot of missing genetics, and I'll come back to that in a second. The important thing here with a lot of these variations is that they're not in the protein coding portion of the genes. These are all in the regulatory regions of the genome. And so they're controlling when, where, and at what level the gene is activated. And it's these sort of regulatory areas where environmental stresses, including things like dietary factors and hormones, actually act. And so there's that part which I'll be talking about in a minute, but also we have this significant issue of missing genetics. And so if we think about those three levels of genetic variation that I talked about earlier, we're talking about population level variance here, and so the thought is that this missing genetics is either family-specific genetic variation, like the Mendelian syndromic forms of clefting, or individual de novo mutations that cause the disease. And so we, just to summarise this quickly, so each person we consider clefting a bit like diabetes and these other common adult conditions, that it is a sort of like, there's a threshold model of susceptibility by which you either have this combination of common and rare genetic variants combined with a variety of environmental exposures which vary from individual to individual, and that's why it makes it so challenging here to work out who's actually going to get the disease. And so we, about 8 years ago, decided to sort of tackle this missing genetics, at least from focusing on these rare and large multi-affected families. And with collaborators, we collected families from across the US, Australia, Colombia, and the Philippines. 85 in total, and these have been pre-screened already for some of the known clefting genes at the time, and we performed whole exome sequencing, so focusing on the coding portions of these genes, those that produce the proteins. We sequenced 3 affected individuals from each of these families, and so far, summarising 8 years of work into one slide, we successfully solved about just over a third of these cases, and so the others have gone on now to whole genome sequencing. It's going to be many, many years before we solve other cases. And so what this has done, though, at least we've identified 11 new clefting genes using this approach. And the important thing, I think, from thinking about a biological mechanism here, that most of those we know from cell biological studies are involved in regulating adhesion and or differentiation of these epithelial cells, these cells that actually line the embryo and actually coordinate those fusion events. And so this is summarised here. So this is what epithelia typically look like. You can see 2 different types of staining here. In this case, if you zoom in on the tight junctions between these cells, you've got a whole collection of proteins that regulate the strength of adhesion and determine cell behaviour and cell shape. And all these proteins here, highlighted in yellow and orange, are all those that we've identified in our families. And so we can pinpoint potentially a shared common molecular and cellular basis for many cases of clefting. And the nice thing here, I guess, also is that from all these cell biological studies over the years too, we know that this complex is very sensitive to things like oxygen levels, mechanical stress, and even nutritional factors as well. So to put it into context, in the embryo, we're focusing on these sort of epithelia here that mediate the fusion. And if we look back at all the, many of the known genes causing clefting, these are all expressed in those epithelia. And same with the new genes that we identified. And we can prove that those epithelia are absolutely critical, because this is an assay we developed a number of years ago in the lab. This is a chick embryo model system where we can actually go in, while it's in the egg still, we can go in and we can genetically manipulate and turn off those clefting genes, those human equivalent clefting genes, just in those epithelia, allow those embryos to keep on developing. And you'll see here, this embryo's got a cleft on the side that was manipulated and not on the side that wasn't. If you allow it to grow out further, where it starts to grow out its beak, you can see the massive cleft here. So there's a bony deficiency, even though it was only disrupted in the epithelium as well. One of the frustrating things, though, and it comes back to this issue of variability, is that as a geneticist particularly, even when we know what and have identified the underlying genetic cause in, say, in one of these families, it's still impossible for us to predict with any certainty whether the child is going to be born with cleft lip or palate or not, or how severe a child will be as well. And that has a lot of implications both for genetic counselling. How do you counsel these families? They know they have a genetic risk. How does that affect your counselling? How do you prepare them for the medical journey ahead when you don't know how severe it's going to be? It also poses challenges for the development of interventional therapies. I mean, you're treating someone who's not going to get a cleft. And then thinking about optimisation of treatment plans afterwards. I mean, we're treating everyone much the same in their treatment plans, although obviously the surgeons modify it as they see fit as we go along. But this could actually impact that as well. So we've moved our research to the mouse as a model system because it now provides a nice in vivo context with this contribution of a maternal in utero environment, just like humans develop. We don't have in the chick system. And so this is just to highlight here that we actually can specifically remove these or disrupt these clefting genes using a particular genetic tool. In this case here, the cells indicated in blue here, which are epithelial cells, are the cells that we're actually removing the gene from, so not everywhere in the body. And this is one example of one of the human genes we identified, the most common gene we identified in our families that's mutated. We mutate this in mice, and we get this beautiful representation, or this full representation of the cleft spectrum that we see in patients. But just to remind you, these mice are completely genetically identical. So you still have this phenotypic variability in genetically identical animals. If we pursue that further, go in more detail, we've looked at lots and lots of embryos now, and the variability in presentation is even more striking, with just a small percentage of embryos that actually appear unaffected. But if we go on and actually look at those embryos, we can see that any embryo that carries that genetic susceptibility is actually still affected. It's just not enough to get a cleft. So with that in mind, you've got this very little bit something that is, in the background there, during that development, is actually contributing to that variability. And so this is actually consistent, this issue of an affected face, even in unaffected individuals. So if you look at parents of children who have cleft lip and palate, even though the parents don't have cleft lip and palate, there's a recognised difference in their facial shape. So they have that underlying inherent susceptibility based on their genetics. And so I've tried to summarise this component here. So there's population level and family-specific genetic variants are really the major determinants of cleft lip and palate susceptibility. But our studies in mice, and particularly the equivalent studies in human monozygotic twins, show that they frequently also don't present with the same cleft phenotype, and some will actually not present with a cleft. And so even in humans, you've got these genetically identical individuals that show massive clinical variability, even when they developed in the same uterus. And so this suggests that there's something about the in utero development that's non-genetic that ultimately dictates whether a child will be born with a disease if they have the genetic susceptibility, or how severe their disease presentation will be. And that's what we've been trying to tackle in our animal models. So what might these non-genetic factors be, or maternal factors be? Certainly evidence from human epidemiological studies suggests that maternal diet is a factor. Also things like alcohol are definitely a non-risk factor, and I'll come back to those too. There's evidence for hormonal fluctuations too. Temperature, certainly in terms of there's more children born with cleft lip and palate who are conceived in the winter months, whether that's related to changes in nutrition or whether it's because parents spend more time in bed in the cold months, who knows. But there's certainly things there. There's evidence that infections or immune activation might actually contribute to risk, as well as environmental toxins. And I think another important thing to consider is also intrauterine position. So where the embryo implants in the uterus, and there's certainly some early data from a couple of decades ago in mice that suggested perhaps the implantation position along the uterus might affect cleft incidence as well. So whether that's an availability of hormones, nutrition, whatever it is, we don't know at this stage. But also there's evidence in human studies that subsequent pregnancies have an altered risk as well. So perhaps some level of intrauterine conditioning is active. But then in humans, we've got the combinations of all of them. So it's really hard and almost impossible to tease apart with humans, and that's why we go to mouse models and the importance of mouse models. So because of this, a lot of those things are not present in an animal colony, and they're hard to necessarily control. And so one of the things we first focused on was diet. And the thing that stood out to us as the ideal candidate here was maternal vitamin A. Now, vitamin A, for those of you who don't know, it's an essential micronutrient that's only obtained from our diet. We don't make it ourselves. This is the biochemical pathway. It's really converted at the end of the day to a compound called retinoic acid. So like folic acid, it basically enters the nucleus, it binds to receptors and activates gene expression across the genome. I'll point out here that alcohol and retinol share a very similar chemical structure, and alcohol is a competitive inhibitor of this process as well, and that's why it's a risk factor. I want to point to this particular protein, which I'll be talking about a little bit in the next few slides, RBP4, so retinol binding protein 4. It is the main carrier protein for retinol around the body. So when retinol is collected from the diet, it gets stored in the liver. RBP mobilises it from the liver, distributes it to all the tissues in the body, plus the embryo as well. And so it's well known from rare cases of severe vitamin A deficiency in humans, but also animal studies when you can inject them with excesses of retinoic acid, that this leads to massive craniofacial abnormalities. And also it's important to consider that vitamin A deficiency is actually quite common in the population too because of food processing. But the question we're actually interested in asking is actually, do fluctuations in dietary vitamin A, but particularly in the normal range, so not these vast excesses or major deficiencies, do they differentially impact an individual that has an inherent cleft susceptibility, a genetic cleft susceptibility? And so I'll come back just quickly, just remind you, retinoic acid, as I said, it binds to receptor complexes and then binds to DNA to activate gene expression. In the epithelia, it actually promotes differentiation and strong adhesion. And in mesenchyme, it has a number of roles, but increases proliferation, but also differentiation at different levels as well. And this is just a 3D image of a mouse embryo in which we've actually got a reporter gene that monitors where retinoic acid is activating gene expression. And so in the blue here is showing at this stage here, at embryonic day 11, it's highly active in that fusion process as well. So the hypotheses that we had were quite simple, is that as individuals with a genetic risk of cleft lip and palate, will they have increased sensitivity to changes in maternal dietary vitamin A? And so when vitamin A is low, we perhaps naively expected that more individuals will present with clefts, and so increased penetrance, but also the clefts will be of greater severity. And so in that case then, effectively in a patient context, that would be increasing the the surgical burden. But then conversely, if we have high or supplemented vitamin A, that then we might predict that fewer individuals will be affected with clefts, and therefore severity also be decreased, and therefore that would have a much more favourable outcome for them from the point of view of management. And so to do this, we have two things we have to control. We have to have an animal that is genetically susceptible to clefts, but also a way to control dietary vitamin A delivery to the fetus. And so in the mice, that's particularly important because mice, like lots of mammals too, can store a large amount of vitamin A in their livers. And so they tightly regulate their levels, mobilising that vitamin A when they need it most to other tissues to try and maintain the levels as much as possible. And so that can last as much as 30 days, and the gestation period of mice is only 20 days. And so we exploited an existing animal model, which is an animal that's deficient in this RBP4 protein. And these animals can still transport retinol to their tissues in the embryo, but only through the minor carriers. And so in that case, they can't pull from their liver stores, and you need extra vitamin A in their diet, so they utilise it up all at once. And so here, about 5 times the recommended daily intake for mice is needed for these embryos to develop perfectly normally and the adults to be healthy. So this is called the sufficient diet. But it does allow us, once we change the diet out, the level of vitamin A in the diet out, you get an immediate response in the serum, and the tissues now are subject to less vitamin A availability. And so the genetic model we use in this case here is, again, an established genetic model. This is the Wnt9b gene model. Wnt9b is a growth factor. It is expressed in the epithelia. It affects the epithelial behaviour, but it also feeds back indirectly to affect growth of the mesenchyme. This model here too shows the same as the previous model I showed you. It shows incomplete penetrance and the full variability of the cleft spectrum seen in humans. I've just represented here the timeline here of gestation in a mouse, so it's 0 to about 20 days, thereabouts. At 11.5 days after the lip is formed, you can recognise all of these mutants. And then by birth here, this is what they look like here, so they still have a variety of clefts. You'll see from looking at the numbers of these and categorising these clefts, about 86% of the embryos have some form of overt cleft, but 14% appear normal. And so they're a good model for looking at sensitivity. And so the experiment is quite simple. We breed these 2 mice together, so we have genetically susceptible animals on this RBP4 deficient background, which we maintain on the sufficient diet, and they're perfectly fine. And then at different stages after fertilisation, so here just around implantation of the embryo into the uterus, or thereafter we switch out the diet either to the low vitamin A diet, so the recommended daily intake for normal mice, or a diet that's about double maternal vitamin A, and then we collect these embryos after the rest of the facial tissues has formed and image them using quantitative imaging. And so really one of the first and really quite striking things we found is as soon as we had the ability to control how much vitamin A was getting to the embryo, we went from 14% normal embryos to 83% normal embryos. And so it just blew me away, and I had to stop and think, so is this really about just the maternal vitamin A, or something else has gone wrong in our experiment? And so we simply put these mice then on a slightly double maternal vitamin A, and all of a sudden we're now getting back to getting lots of clefts again, which is a little bit surprising because we thought we'd actually get suppression there. But what this actually tells us is that there's an optimised level of maternal vitamin A that's actually required to actually significantly reduce the penetrance or expressivity of clefting in cleft-susceptible individuals. We can quantify this as well because all our imaging is fully quantifiable. And so if you measure, for example, the width of the lip region as a severity measure, this is what they normally look like, these Wnt9b clefting animals. They're all very severe. On this controlled diet here, the ones that do have clefting are very, very mild. On the high diet, you start to see severity increase as well as penetrance. This is what they look like as a whole in terms of the variability. So these are genetically identical animals. There can be a bilateral cleft lip and palate, bilateral but partly fused, different amounts of unilateral. And if you look at these same embryos and their craniofacial skeleton, these 3 animals on the top, you see complete separation, all their bony skeleton down the midline, they all have cleft palate. And you can also see the variable impact on the primary palate, the alveolus where the teeth are formed as well. So a huge variability, and when we add that and maintain that vitamin A, optimal vitamin A, we're pushing this whole spectrum to the left. So one thing I want to point out here, this is I said these Wnt9b animals, when we don't control diet, said all of them appeared to be abnormal early on in development, but by birth we had 20% that looked normal. So we had this inherent ability in utero for 20% to repair. The analysis I showed you before is all at this embryonic day 15.5, so about 4 or 5 days before birth, when we've got now in the optimised diet, nearly the complete opposite. We've got only 20% affected, thereabouts. And so the question is, by birth now, so now we're going back looking closer to birth, and what's the percentage there? Have we actually fully recovered nearly all of these animals on the same trajectory that we had the other animals? And just to show you here, this is one of those animals, close to birth, that appears pretty much normal there, but if you look carefully, you have 2 little notches just on the lip there. So it has slightly delayed fusion, but clinically, if this was a patient, they'd be perfectly normal and would not have surgery. So there's promise that here that we're getting close to almost full recovery for many animals. So what happens on a low diet? We had to actually do this diet switch to the low diet actually only 2 days before the lip forms because it was so severe. But it affected all embryos where we're controlling that dietary availability. But in this case here, just 2 days before, we actually surprisingly saw an increase in clefting as well. But the cleft presentations in these animals are very different. The faces are much wider, the clefts are more severe as well. And so higher vitamin A and really low vitamin A impacts facial development, but in different ways. And so I think from a clinical point of view too, I think we need to think about what the presentation is in families too, to decide whether vitamin A increase or decrease might be more effective. And so one thing I also mentioned earlier, that human cleft lip and palate is well known that left-sided unilateral clefts are twice as common as right-sided. Hadn't been reported in animals before. In our characterisation of this model, we also found that these mice have a 2-to-1 left-sided bias. And even more interesting is that when you put them to the higher vitamin A diet, the left-sided bias now increases to 3-to-1. And so diet is actually impacting the sidedness of the embryo as well. We have a good hypothesis, a good thought as to what's actually underlying that mechanism, and I think it's the early establishment of left-right asymmetry for your internal organs that's contributing to that. And just to prove that this is not just because we're on this really fine control of vitamin A, we went back and did just the regular cleft susceptible mice, not on the controlled diet, the tightly controlled diet availability, and put them on the different diets. And also there was a strong correlation with the level of vitamin A in the maternal diet and left-sided preference for unilateral clefts. And so I summarise here, just in conclusion here, that I really want to emphasise that cleft lip and palate risk or susceptibility is dictated primarily, in my feeling, by a combination of population level variance, but also more importantly, rare or family-specific genetic variations with higher biological impact. And so at least in 35 or a third of families with a really strong family history, we can actually identify those changes because they're in the coding region. But those other families, there's probably in regulatory regions that are much more challenging to identify. And also that maternal dietary factors, I think, are going to, either in isolation or in combination with all those other potential contributors, may have a really large impact on whether genetic susceptibility ends up producing a cleft in an individual. And so apart from vitamin A, obviously we're now also interested in alcohol and the interaction between that and vitamin A. Fats and cholesterol, which help metabolize vitamin A, is really important too, but also other factors, including hormones and hypoxia conditions and metabolic rate. And then I think an interesting thing from the perspective of humans, which is a little bit more challenging to identify in mice, although we've just got some funding to start to deal with some of this, is looking at modelling some of these regulatory variants in humans in mice as well, and to see whether they're going to have a differential effect in response to things like diet. And then at the end of the day, if we come back to what I started with, is this issue of folate and the example of folate, which has been so incredibly successful, that's still not very effective for about a quarter of those genetic causes. And so the question here is, are all genetic susceptibilities in humans also responsive to vitamin A? And so we need a lot more work goes on before we even think about whether we should be applying this in the clinic, because there's a lot of apprehension about vitamin A in general, because there's recommendations not to use really high vitamin A creams for skin creams, for example, during pregnancy because of the risk of birth defects. But have to remember, these are exceptionally high levels of vitamin A. And what we're trying to do here is trying to essentially provide optimised nutrition for people to minimise that risk instead. And so that's the distinction here. And I'll finish it up there, leaving you with one final thought. And this has come from I had the privilege of attending a World Health Organization meeting many years ago, where the topic was on the prevalence of these craniofacial malformations, particularly in third world countries, and how to deal with it, particularly when they don't have access to the skilled surgeons, healthcare, or even financial resources. And so I'm going to leave you this question, which is sort of like the ultimate dream here. And so imagine if we could predict an individual's risk of being born with clefting based on their genetics, even before they're conceived, so based on parental genetics, and then intervene with simple dietary or lifestyle modification to prevent it. And I'll leave it there. Thanks for your attention.
So we'll open it up for questions. If you have a question here in the room, you can raise your hand, and Anna or Faresh will bring a microphone to you. We'll also be watching for questions online via Teams, so we'll interrupt occasionally for one of those. Questions from the room?
Wonderful talk, Tim.
Thanks,
Thank
Paul.
you very much. I wondered if you had tried to rescue the phenotype. Do you know that if you give the embryos reduced diet in the first couple of weeks, or a couple of days actually in mice before the lip is formed, and then later supplement the diet, are you able to rescue the phenotype?
Yeah, it's actually a really good question, because originally we thought that we're trying to optimise that contact between the processes. But I think the evidence from this sort of inherent ability of mice, a small percentage of them to continue growing, and mice may be a little bit different to humans here because they have that extended growth period of the facial process because they've got this elongated snout. And so I think there is that possibility that even a delayed thing might help bring those facial processes closer together, even if they don't get proper fusion. Even that sort of improvement would minimise surgical intervention, I think. And so, but we haven't actually done that experiment yet, but it is something that would be really worth doing.
And just related, in the embryos, with the numbers that you saw, a reduced or increased number of clefting earlier and then a decrease later, that was not due to embryo death, obviously. Is there any evidence that during pregnancy, retinal supplies from the mother can actually increase during pregnancy and maybe cross the placenta into the embryo, artificially?
I don't think that's really well known. We've spent quite a bit of time this last year developing a mass spectrometry-based assay to quantify retinols in maternal serum, but also in embryonic tissues. And so we're at that stage now where we've actually got that as a really robust technique. And so when we change that maternal diet, we can actually see how that responds. And so in addition to that is doing studies like watching to see on the maternal use, the availability for the serum during pregnancy, up to and during pregnancy as well, would be a valuable thing to do. Yeah, there's a lot of factors I think that we have to really be aware of before jumping in too wildly to something more significant. So,
thanks so much. That was great. My question is there any specific growth factor that you suspect is being modulated by the vitamin A levels?
Yeah. So things like the Wnt signaling pathway are significantly regulated by vitamin A. And so it may have been somewhat fortuitous that we chose the Wnt9b model, but I think Wnt9b also has a role in remodeling junctional complexes as well. So I think there'll certainly be a pathway there that my suspicion will be responsive to vitamin A. And Wnt9b also initiates a number of other expression of a number of other growth factors as well. And so I think there's a cascade there. And so from the human genetic studies, some of the genes, there are growth factor pathways that have been implicated as well, and they're things that are certainly downstream of the Wnt signaling pathway. So I don't think it's just one growth factor pathway, but I think it's many. But there's a subtype of clefts that present, and you may have seen it in my variability slide, there are some kids that have what's classified as bilateral or unilateral cleft, but their medial tissue is very, very small. And I think they're actually a different genetic mechanism. I think they're a cilia-based problem, but they're classified perhaps incorrectly as classical cleft lip and palate, and there's a minor form of a midline cleft, and they will be very different. And we actually have a model in the lab of that type of presentation, and we're currently doing the same experiments to see whether they respond in the same way. But thank you for the question. Yes, Dan.
Thank you. That was a great presentation. So I was thinking about all the activity with the morphogenesis that's going on. Does that generate a lot of free radicals?
Oh, now you're going to be pushing me.
No,
I don't know.
The reason why I'm asking is that, if you look at the structure of vitamin A, they're antioxidants, right?
Yeah.
And one of the interesting things about antioxidants is when they have an appropriate abundance, they can have antioxidant qualities, but then when they get overabundant, they can actually promote free radical formation.
Yep. Yeah.
And I don't know if you have an idea for the mechanism of why overabundance of retinol leads to reformation of the cleft, or however.
Yeah, I mean, it's a good point. I mean, I know retinoic acid or vitamin A pathway has a big impact on metabolism in general. And I think there's plenty of studies done in cells within the lab and also some animal model studies. Different levels of vitamin A can have very different effects in terms of gene expression. So once you get past a certain level, you'll now activate a whole different set of genes as well. And so I think we run into this complication here trying to interpret severe, really high doses, really low doses, and trying to make generalizations about them. But I think there's a lot more subtlety in whether things like metabolism, free radicals, things like that actually have an impact as well. I don't think anyone's really looked at it seriously, but I think we have the capacity to now with some of the mass spec approaches that are getting so sensitive to see what changes are there. Again, I don't know what impact they would have, but it's certainly an interesting way to go. So thank you.
I enjoyed the talk. Thank you. I have a question about the retinoic acid receptors. If I understand them correctly, they don't really work by themselves. They work a lot of times with as heterodimers with other transcription factors.
Yes.
That's another level of complexity that may need to be looked at as well. What are your thoughts on that?
Yeah, there are. I mean, the retinoic acid receptors, you rightly say, they form heterodimers, they do complex, and they're actually they're considered to be almost a pioneering factor in a way. When they bind to the DNA, they start to recruit all the other complex involved in activating the gene. So there is a lot of complexity there, and I think that's another 20 years of work to look at. But I agree. I mean, when you look at some of the transcription factors that have been shown to be mutated in cases of cleft lip and palate 2, they are often some of them are the early pioneering factors as well in epithelia, like P63 as well. So that's one of the early ones that's recruited there. So there may be some role there just with the early initiation of a transcriptional pathway that defines epithelial differentiation, for example, but it will be a complex set of events that are going on. Thank you.
I think we just have a few more minutes, so I have a couple questions I've been really wanting to ask. And so, one question I had, are there any clinical scenarios where genetic testing is indicated or used, or is it just purely on a research basis?
These days, I think a lot of kids that come in with isolated clefts don't even get genetic testing because I think the hit rate, certainly from our studies, the hit rate is less than 5%. And so we have a panel of 60, 70 genes from human studies and implicated from mouse studies, and people focus on them, and the hit rate is so low that they don't even bother yet. And I think this is where I think it's going to get complicated, having to look at polygenic risk factors where we're looking at the spectrum of common variants that people have and seeing, trying to do studies with that, which is going to be challenging. When you've got a family history, they're way more likely to go get genetic testing. At this point in time, though, it's really just for genetic counselling purposes. And I think in some respects, depending on the gene involved, we may expect other features potentially to present as well. And so it helps with some of the planning for the surgical process that's going to come later on. But that's about it at the moment. So I think we need a lot more done.
Next question. I think the variability is really, really interesting, both when you intervene with the dynamic range of the vitamin A as well in just your model. So have you looked at exploring transcriptomics and epigenomics in spatial genomic expression across the face?
Yeah, we just got funded last year for this project after we've completed 6 years of animal work. Now, when that will actually fund some of the transcriptomic type work and the spatial work. So, yeah.
Beautiful. Can't wait to see that follow up. Any other questions?
Do you see that variability in the chick embryos, or maybe you can control more of the variables?
Well, the chick embryo is a little bit different because in that case, we're actually using electrical current to introduce and to interfere with the genes in that. And so there's a lot more variability, technical variability that goes into that. So it's really hard to say, but thanks. Yeah.
So I had a small doubt. In your conclusion, you mentioned about hypoxia. So have you looked into the HIF pathway? Because I believe vitamin A binds with the...
with HIF1 alpha as well, yeah, it does. And there's a number of ways hypoxia can have an effect. So hypoxia also regulates a couple of proteins that have a major role in regulating epithelial adhesion strength as well. So there's that role, which is thought to probably be the more prominent one, but certainly from the transcriptional point of view, interaction with HIF1A, again with that receptors and the activation pathway, could also be another component, but we haven't looked at it yet.
So do you think the HIF inhibitors already in the trials can be used here?
It's possible. I don't know whether you'd want to use the inhibitors. I think that might be worse.
Yeah.
But certainly as a modifying thing, that would be interesting. Yeah.
Thank you.
Dr. Cox, so thankful for your talk and delivering in a way where it's accessible and the impact that you're having here in the state of Missouri and beyond. So as a token of our appreciation, we would like to share this plaque and join me in thanking Dr. Cox again.
Thank you.